How do antibodies use opsonization in your immune system? Have you ever wondered how your body knows to attack harmful invaders? One of the key players in this process is antibodies, and today we will focus on how they use opsonization. Opsonization is a fascinating mechanism that helps your immune system identify and eliminate pathogens. Antibodies are Y-shaped proteins produced by your immune system. They specifically recognize antigens, which are unique structures found on the surface of pathogens like bacteria and viruses. The tips of the Y-shaped antibodies, known as the Fab regions, bind to these antigens. This binding is essential for what happens next. Once the antibody attaches to a pathogen, the other part of the antibody, called the Fc region, becomes exposed. This is where the magic of opsonization happens. The Fc region can be recognized by receptors on immune cells such as macrophages and neutrophils. These immune cells are like the body's cleanup crew, ready to engulf and destroy invaders. When antibodies coat a pathogen, they essentially mark it for destruction. This tagging process enhances the ability of immune cells to identify and grab onto the pathogen. Think of it like putting a bright sticker on a package to make it stand out. The immune cells can then attach to the pathogen more effectively, leading to a process called phagocytosis. Phagocytosis is when immune cells engulf and digest the pathogen. The Fc receptors on phagocytes bind to the Fc portion of the antibodies, acting like a handle that helps them grab the pathogen. This makes the entire process of destruction much more efficient. But that's not all. The interaction between the Fc region of antibodies and immune cells can also recruit additional immune responses. This can include the releases of toxic substances that help kill the pathogen. Antibodies also work with a group of proteins known as the complement system. When antibodies bind to a pathogen, they can activate these complement proteins, which further coat the pathogen and promote inflammation. This teamwork amplifies the immune response, making it even more effective. Among the different types of antibodies, immunoglobulin G subclasses 1 and 3 are particularly good at opsonization. They bind well to Fc receptors on phagocytes, making them highly effective at marking pathogens for destruction. When these antibodies coat encapsulated bacteria, which are usually harder for immune cells to engulf, they make these bacteria easier targets. In summary, antibodies use opsonization by binding to pathogens with their Fab regions and exposing their Fc regions to immune cells. This process flags pathogens for destruction, helping your immune system clear infections more rapidly. Opsonization is a vital part of how antibodies contribute to your body's defense against harmful invaders.